Hi, welcome to the lecture on human brain. The human nervous system can be viewed as a three-stage system. As you can see in the block diagram, central to the nervous system is the brain, which is represented by a neural network, which continuously receives information, perceives information, and makes appropriate decisions. You see the two sets of arrows shown in the figure. Arrows pointing from left to right indicate transmission of information bearing signals through the system, and the arrows pointing from right to left signify the presence of feedback in the system. The receptors here convert stimuli from human body or external environment into electrical impulses that convey information to the neural network, which is nothing but the brain. The effectors convert the electrical impulses generated by the brain into visible responses as the system outputs. Let us have a comparison between the brain and the computer. We will just have a basic information about the neurons in our brain. We know that the neurons are the structural constituents of the brain, and also neurons are 5 to 6 orders of magnitude slower than the silicon logic gates. That is, generally the events in a silicon chips happen in a nanosecond time, whereas the events in our brain happens in a millisecond range. There are 10 billion neurons and 60 trillion synapses are present in the human cortex. If you come to the energetic efficiency, so the brain has 10 power minus 16 joules per operation per second, whereas the computer today is having 10 power minus 6 joules per operation per second. So if you compare all these things, you can come to a conclusion that the brain is incredibly slow in comparison with the modern computer processes. That is, a neuron in the human brain runs 200 operations per second, whereas a transistor, which is a basic building block of a 2 gigahertz processor, which we generally find in our smartphones, runs 2000 million operations per second. So this in turn says that this makes the man-made transistor a billion times faster than the biological neuron. Next are the synapses. Synapses are the elementary structures, and these are the functional units which mediate the interactions between neurons. If you see in this diagram, so we have synapses here. So these are the synapses. They mediate the interaction between one neuron and the other neuron. When we talk about the synapses, the most common kind of synapse is the chemical synapse. Let us see how this chemical synapse is going to operate. So a presynaptic process liberates a transmitter substance, and this transmitter substance diffuses across the synaptic junction. So this synaptic junction acts as a connecting junction between these neurons, and this particular process which liberates this substance, it acts on a postsynaptic process, and later the synapse converts the presynaptic electrical signal into the chemical signal and then back into the postsynaptic electrical signal. Here you can see these are the dendrites of the neuron. This is the cell body, and this is the nucleus, and this is the axon hillock, and this is called presynaptic cell, and this long path between these two, that is this long tail like structure, is called axon, and this is called myelin sheath. It is made up of myelin sheath, and this is the signal direction from one neuron to another neuron, and these are the synaptic terminals, and this is the synapse. Okay, and this is called the postsynaptic cell, and now this is the communication direction from this particular cell to the other neuron cell. So if you see here, this is how actually a biological neuron is going to look like. So this is the neuron. So these are the dendrites, this is the axon. So if you see this, the electrical impulse is going from this particular place, that is the electrical impulse is passing through the axon from one particular neuron to the another particular neuron. So here when you say this, These are called the neurotransmitter molecules, and these are called the receptors, as we have discussed in our block diagram about the receptor, right? So this is the receptor and these are called the synapses. So in an adult brain, plasticity is accounted by two mechanisms. So we have already discussed about what is plasticity in our previous lectures. So there are two mechanisms. The first mechanism is creation of a new synaptic connection between the neurons. Okay, and the second one is modification of the existing synapses. As we already discussed that, so brain while learning new things, it is going to adapt to the changes. Okay, so this plasticity is accounted by either the creation of the new synaptic connections or the existing synaptic connections may be modified within the brain. So generally neurons come in different variety of shapes and sizes in different parts of the brain, but the most general or the most common type of cortical neuron is a pyramidal cell. So this is an example of a pyramidal cell which you are seeing here. So the pyramidal cell receives most of its inputs through the dendritic spines. So if you see here, these are the dendritic spines and these are the synaptic inputs and these are the segments of the dendrite. Another fact of pyramidal cell is that it can receive 10,000 or more synaptic contacts and it can project onto thousands of the target cells. So here these are called apical dendrites, these are called basal dendrites, and this is cell body, and this is axon, and these are synaptic terminals. So now let us see how neurons are going to encode their outputs. So generally neurons encode their outputs as a series of brief voltage pulses. So these voltage pulses are nothing but they are called as action potentials or spikes. So now you see here this is called an action potential. So this is the potential through which these neurons are going to encode their outputs. So these action potentials originate at the cell body, okay, and then they propagate across this axon and they propagate at a constant velocity and amplitude also. So we can consider this axon as the transmission lines, so which transfer the information from one neuron to another neuron, and if you can see the dendrites, so these are the dendrites, so these are called the receptive zones of the neuron. So here comes the structural anatomical organization of levels in our brain according to Churchland. In our brain, there are both small scale and large scale anatomical organizations where different functions takes place at different levels. That is, at lower levels, there are some different functions, and at higher levels, different types of functions takes place. So when you see here, the most fundamental level of anatomical organization comes with the molecules, and from the molecules, we have synapses where the synapses depends upon the molecules or ions for their action. In the next level we have neural microcircuits and then the dendritic trees, and then in the next level we have neurons. The neural microcircuit refers to the assembly of synapses organized into patterns of connectivity to produce a functional operation of interest. So a neural microcircuit can be taken in analogy with a silicon chip which is made up of an assembly of transistors. The smallest size of microcircuits is measured in micrometers, whereas their fastest speed of operation is measured in milliseconds. So these neural microcircuits are grouped to form dendritic subunits within the dendritic trees of individual neuron. So the neurons, which are 100 micrometers in size, contains several dendritic subunits. So in the next level we have local circuits, so which are 1 millimeter in size, wherein these local circuits are made up of different types of neurons. Then followed by the interregional circuits, and then comes the top level, the central nervous system. Here in this figure we can see the cyto-architectural map of the cerebral cortex, which is given by the Brodmanns. This figure clearly shows the different sensory inputs are mapped onto the corresponding areas of the cerebral cortex in an orderly fashion. You see here areas 1, 2, 3 represent primary somatosensory cortex, area 4 represents primary motor cortex, and here also it says the primary motor cortex. Areas 44 and 45 represents Broca's area, and area 22 represents primary auditory cortex. So in this manner, different areas represent the different types of sensory inputs mapped to the cerebral cortex. So in this lecture, we discussed about what is human brain and then the comparison between the brain and the computer, and we also discussed about what are synapses, okay, and the biological neuron structure, and then we have discussed about what is a pyramidal cell, and then we have discussed about what is action potential and how it is being propagated, and in the last we discussed about the structural organization of levels in a human brain. And then this is the Brodmann's cytotonic map. Okay, thank you.